Okay, so in today's class, we'll talk about chromosomes, how were they discovered, what is their structure, and how many types of chromosomes are there. So, dear students, chromosomes were accidentally discovered by a German embryologist whose name was Walter Fleming. What was he doing? Actually, he was observing the salamander larvae after treating them with Perkins aniline dye. So during his observation, he found certain dark colored structures within the cell. So he named them as chromosomes, means colored bodies. But to your surprise, they are actually not colored. They are colorless bodies. So what are the important characteristics of chromosomes? They are actually thread-like structures. They are specific in number. For example, in case of human beings, in case of Homo sapiens, we are having 23 pairs of chromosomes, and this is fixed in every human being. Okay, and the chromosomes are made up of chromatin materials, and they contain genes which bear certain traits for a certain feature, for certain characteristics. If you look at the composition and structure, first we will discuss the structure of chromosome. Like chromosome has got two chromatids. Since these are the chromatids of the same chromosome, so they are termed as sister chromatids, while in case of homologous chromosomes, in which one chromatid is received from the mother cell and the other from the father's cell, so they are non-sister chromatids. But here that you are looking on the screen, this is basically the chromosomes that is made up of two sister chromatids. You can see here that in the center of the chromosome there is a spherical area or the spherical object. This is called centromere. With this are attached two arms. One is P arm and the other is Q arm. The P arm is smaller in length as compared to the Q arm. Okay, now if you look at the composition of chromosomes, then chromosomes are composed of DNA, histone proteins, and RNA also. Here you can see easily that DNA is in turn composed of deoxyribose sugar, certain nitrogenous bases like adenine, guanine, cytosine, thymine, and beside this they are also having phosphoric acid. So this gives the property of acidic nature to DNA. On the other hand, histone proteins are basic in nature. Okay, so basically histone proteins are made up of very basic polypeptides which are rich in the amino acids arginine and lysine. And 8 of these histone proteins, what do? They form the core, okay, and that is called the nucleosome. And around this nucleosome, the DNA coils itself. Being acidic in nature, it gets attracted towards the basic histone proteins. So hence it makes a very intact structure, and that's how it makes the chromosomes. And you know that DNA is a double helix structure. It contains double strands. Okay, so if you look at the types of chromosomes, then the chromosomes can be divided into 4 types, right? One is metacentric, the other is submetacentric, acrocentric, and telocentric. These are actually divided on one important base which is called the position of the centromere. So as you can see, in case of metacentric chromosome, this centromere is lying in the center. Okay, in submetacentric, the centromere's position is off the center, like it is not towards the center but it is off to the center. In case of acrocentric, the position of the centromere is subterminal, and in case of telocentric, it is terminal. For your knowledge, I want to tell you here that telocentric chromosome is not found in human beings. Okay, so I hope that you understood these types of chromosomes. How is the structure of the chromosome? What is its composition? What are its characteristics? And who discovered the chromosome? So that's all for today's class. I will catch you guys with another topic in the next class. Till then, take care.